Hello, everybody. Welcome to NeuroLingo installment number 3, and hopefully there will be many, many more after this one. Thank you for joining us this morning. What an absolute treat. My name is Jeremy Ullman. I am a graduate from McGill University 2018, Go Class of 2018. I am a podcast host, and I'm happy to bring my services here to NeuroLingo for the day. NeuroLingo aims to make neuroscience research accessible, understandable, and engaging to you regardless of your academic scientific background. Today, we're going to be having 3 neuroscience graduate students from University of Montreal who will be presenting their own research at the master's and PhD levels. Our featured speakers have been preparing their talks for the past 3 months and are extremely excited to share their work with you, and I'm excited to engage with them as well. So after each talk, we're going to have a quick Q&A session, and we ask that you please use the chat to ask questions to the speakers directly. Just so you know, there is a $40 giveaway after each presentation to an audience member that's you who asks a question. So don't keep any of those great ideas and thoughts inside. Share them with us for a chance to win. The winners will be selected at random and will be contacted after the event by email. Okay, so keep your eyes peeled for that. Now, without further ado, let us hop into our first presentation. We are joined by Hiba Dagar, very special. She's a PhD student at University of Montreal, and her research is focused on personalized medicine and its potential for identifying genetic causes for rare diseases, opening up new treatment options for targeting these genes. Themes in her talk include, but are not limited to, personalized medicine, worms, genetics, rare diseases, and novel treatment programs. So let's welcome Hiba Dagar to the virtual stage. Welcome back. Thank you, Hiba, for that excellent talk in the past and now for being here in the present. Thank you for unmuting as well. So, quick question. What drew you to doing research on rare diseases, knowing that there are fewer people who have rare diseases, so you have the potential to help fewer people? How do you reconcile that?
Yeah, thanks, Jeremy, for your question. So first of all, the rare disease community is a group of different diseases that, yes, of course, involve few people at a time, but when you merge them together, it's a lot of patients. It's actually millions of people in this world. According to the CHR, they have done an assessment, and actually around one child on 15 is born with the rare conditions, and that's more children as the number of children that are born with cancer. So if you group them, it's not that rare. And obviously, because these genetic conditions are often associated to children because they were born with these conditions, there's an urgent need of therapy, and we couldn't just abandon them and not treat them. It's actually, it pushes the research to be innovative and try to find low cost research. Obviously, we couldn't think the classical way of waiting 10 to 15 years to have a drug approved and tested in many animal models. So using these drug repurposing or small models are really cheap and easy to maintain. We can have these really personalized avatars or animal model for all the genetic conditions that theoretically could exist in this world, and so we have these patients to be involved in the clinical field and not feel discriminated, obviously.
I think it's great that you're using these animal models. Specifically, you mentioned C. elegans, this worm. We actually got a question in the chat. Somebody's asking if there are other organisms that would be well suited to the study of personalized medicine, different from these worms. Have you worked with anything, or do you know of any research that uses other kinds of living beings?
Yes, that's a great question. Thanks for the person who asked that. So usually we start with worms because of the easy, all the features I mentioned in the talk. But there's also another model that is a fish model, which we call a zebrafish. Usually we know research is associated with mice or humans and cell cultures, but there's also another genetic tool, which is zebrafish, which is kind of in between the mice and the worms. So they're really easy also to maintain. And you have a phenotype. So you have clinical features that appear really early in the development of these fish. I work actually on fish models as well for my projects. So we do have the C. elegans models, but also the zebrafish models. So we can push the research a little bit forward and try to validate the things we find in worms in fish also.
Totally. I love that you have that kind of reciprocal, two-sided approach, because you don't want to just try one method. So by being able to use both of those, then I guess, yeah, you can kind of cross-reference.
Definitely.
So that's great. So you didn't have time to get into a tremendous amount of detail during the talk, but I'm just curious, could you maybe quickly summarize how you actually model and mimic rare diseases in these worms?
So that's, we have to thank the genetics and the innovation again in research. In the past few years, there was a Nobel Prize that was given to researchers who invented the CRISPR-Cas9 technique. And that's a genetic, it actually revolutioned the genetic field because we are able to mimic really specific mutations, and by changing directly the DNA of any in vivo model that exists. So what we do is that we take the sequence and the mutation that is in the patient with whatever condition the person has, and we were able to change the exact same nucleotide or to mimic the exact same mutation in the DNA of any living model we have, and that changed the field in many aspects. So yeah,
that's awesome. So technically, we could do genetic mutations on humans using this technology as well.
I mean, that was one of the ethical issues that people start talking about, because we actually can do it technically, and there's some regulations that have to be done to set, the use of such technologies and not, try to modify everybody's DNA. So there's an ethical issue related to that, but the technique is there for sure.
Wow. Amazing. Well, I look forward to hearing more about your research as you go. How much longer do you have left in your degree?
I officially started last year, so I'm in the middle of it. We'll see how it goes. I'm looking forward as well to see where this project goes as well.
Wonderful. Well, good luck. I'm also looking forward to it. So we'll keep in touch. Thank you so much, Hiba. We're going to head on to the second presentation for the day. I will catch you later. Hiba is going to be in the chat answering more of your questions if you have them as they come. So please do continue to send those in. Awesome. So getting ready for talk number 2 with Stephanie Coronado Montoya. She's a PhD candidate at University of Montreal, once again. Her research focuses on a cannabis harm reduction program for people with psychotic disorders who use cannabis. Themes in her talk include, but are not limited to, cannabis, psychosis, empowerment, and harm reduction programs. So let's hop into talk number 2.
Did you hear the good news? Well, it's old news now, but 4 years ago, Canada decided to legalize cannabis for recreational use. Now, this was a fact that was celebrated by so many people and in very, very creative ways. And with reason too, because for a lot of people, cannabis use was a fun time. Well, I'm here to tell you the bad news. The bad news is that for certain groups of people, like people with psychotic disorders such as schizophrenia, cannabis use can actually be very problematic. My name is Stephanie Coronado Montoya, and I'm a PhD candidate at University of Montreal. My research, which spans across Canada, focuses on finding solutions for people with psychotic disorders to manage their cannabis use in a safer way. Now, just so we're on the same page, people with psychotic disorders tend to experience symptoms such as hallucinations, delusions, paranoia, lack of motivation. Cannabis use in people with psychotic disorders has been a problem without a solution for decades now. And today, I'm here to tell you about the interesting solutions that my team and I are proposing to address this problem. And interestingly, some of these solutions might pertain to your everyday life. But first, how bad can cannabis really be? Well, for people with psychotic disorders, it turns out it can be pretty bad. So whether we're talking about mild use or very frequent use, the fact is that the consequences associated to them are pretty serious. We see people that use cannabis also have longer hospitalizations and more frequent hospitalizations, worsen psychiatric symptoms, worsen suicidal ideation, worsen day-to-day living. This is nothing that sounds like fun. Not just that, but in terms of prevalence of cannabis use, while we see a prevalence of cannabis use in the general population of about 25 to 40%, in the population of people with psychotic disorders, this number jumps up to 60%. In terms of problematic cannabis use, in people with psychotic disorders, 40% of them have a cannabis use disorder. Now, not just that, but there are also very few programs that are adapted for this population to help them with their cannabis use. I did a review of all the data up until 2019, looking at how many cannabis preventive interventions or programs had been tested for people with psychotic disorders. And you know how many I found? 4. Only 4 programs had ever been tested for this population. So what can we do for them? Well, there's the most traditional approach, which is called abstinence. And abstinence, in its most extreme forms, essentially means just say no to drugs. So while theoretically this is a great idea, there are a lot of people who are not interested in quitting, who cannot quit, or do not feel capable of quitting just yet. So what can we do for them? Well, there is another approach, and it's called harm reduction. Now, harm reduction doesn't require a participant to stop using the drug completely, nor does it encourage the person to continue using the drug. However, harm reduction works with a person who's already using that drug and does not feel capable or not willing to stop their drug use, and they work on goals that will help them to reduce the harms associated to that drug use and which will ultimately help them improve their well-being and their quality of life. So for example, if a person is using illicit substances and is often getting into legal problems because of that, and that is their main concern, they do not want to quit. A harm reduction goal would be to encourage this person to use the illicit substance in more private places where they have less risk of legal problems. Not because we want to encourage the substance use, but because we want to reduce their risk of harm. Now, some of you might be saying that this harm reduction idea is way too radical, or why are we even talking about this? Why even promote it? Why not just abstinence all the way? Well, as it turns out, harm reduction can actually be a very effective strategy to deal with risky behaviors. And we actually see many examples of this in everyday life. Let's talk about sex. A lot of people know that having sex can also come with the risk of sexually transmitted infections. But do you see people abstaining from sex for this reason? Most people prefer the harm reduction alternative of using condoms or getting tested. There's also cars. Cars come with a risk of serious injury, but do most people prefer to walk or take horses to work? Most people end up using a seatbelt. Now, in the addiction field, there's also other examples that are well established and supported by data. For instance, intravenous drug users, instead of abstaining from drug use in the case where they don't want to, or they're not willing to, or they cannot just yet, they also run the risk of having infections because of unsterile needles. And as a harm reduction alternative, they go to supervised injection sites. Well, what about cannabis harm reduction? There isn't much in that realm, but that's where my team and I come in. My team and I are working on a cannabis harm reduction program for people with psychotic disorders who use cannabis. And this program is called CHAMPS, the Cannabis Harm Reducing App to Managing Practices Safely. CHAMPS is a short online program developed for a mobile app that aims to teach participants about the intersection of cannabis and psychosis and its consequences, as well as teach participants about different harm reduction strategies they can use for cannabis. Then, they ask participants to question themselves, why are they using cannabis? Why do they want to stop using cannabis? We also teach goal-setting skills. The ultimate aim of CHAMPS is to actually empower the participants by giving them both the knowledge and the skills for them to create their own positive changes in terms of their cannabis use. Now, CHAMPS is the first of its kind for the population, and I do want to tell you about some very interesting features that are built into it. The first one that I want to tell you about is that, like the United States, CHAMPS is built by the people, for the people. And what I mean by that is that CHAMPS was built as a collaboration between researchers, clinicians, psychologists, and very importantly, patients. Now, why is that important? Well, traditionally, clinicians and researchers built programs for patients, but not necessarily consulting them, but rather consulting the literature or best practices or using their field expertise. However, patients have very valuable lived experience, and they can tell us about their realities, their needs, and their wants. We see in programs that are developed for people with psychotic disorders very low participation rates and high dropout rates. So perhaps by consulting the patients directly, we can address this problem as well. That's what we did. We created a CHAMPS committee of people with lived experience, and we asked them for feedback from the beginning of the app development and the content to the end. We asked them their opinion on the content we were creating and the tone of the app. We asked them for what kind of information they would have, and they would tell us, very honestly, when it sucked. And they would also tell us what features they liked and what features would be helpful. One of the best participants' feedback that we got was one person who enjoyed using it so much because she was just swiping left and right like it was Tinder. She was having a great time, but actually, at the same time, we were educating her about harm reduction strategies. The feedback that we got from participants, from that patient voice, really helped us to mold CHAMPS for people like them to respond to their needs, their desires, and their realities as well. Now, another really cool feature about CHAMPS is that it's developed on a mobile app, And given that this population has low levels of motivation, it is really advantageous for them to have an easy-to-access program at their hands. And finally, that Canadian cherry on top, CHAMPS was developed in French and in English. That way, more people can access that help that they might need. So, CHAMPS is now fully developed and it's in its testing phases. And because it's the first of its kind, we cannot first test for effectiveness. First, we have to do what is called a proof of concept trial. And essentially, we'll be looking at the acceptability and feasibility of both CHAMPS and running a clinical trial with this population. To do this, we have to recruit 100 people with psychotic disorders who use cannabis across Canada. And we will be directing them to either CHAMPS or treatment as usual. We'll be collecting data at the end of the trial to see how many people completed CHAMPS, how acceptable it was, how many people completed the study, and all of its measures, in order to better understand the acceptability and feasibility. We'll also be collecting other exploratory data to better describe how CHAMPS is affecting its participants. We'll be looking at cannabis use and their use of harm reduction strategies and other variables just to better understand how CHAMPS is working. Now, whether or not CHAMPS works, my hope is that the research we're doing will expand the amount of options we have available to people who want to use cannabis, but in a safer way. That it not just be abstinence all the way, but we can also talk about harm reduction. As many know, that legalization of cannabis use was great news for many people. And my hope is that the work we're doing will help turn that bad news for some into good news for all. Thank you.
Okay, welcome back. Thank you so much, Stephanie. You remind me why I quit my first master's degree. It's because I cannot compete with that. No point. So I want to start you off with a quick question from the chat, which is, first of all, thank you very much for sharing some great arguments for harm reduction. Sounds like a reasonable kind of program to set up, but is it applicable to those suffering from psychosis who are also homeless?
So assuming that's a very good question, actually. And assuming we're talking about their cannabis use for homeless people who also have a psychotic disorder, I think even though this is an even harder to reach population because they're more vulnerable, they're living with more obstacles, more barriers, things like that. Despite that, if we can't get them into treatment and they do need treatment, then yes, harm reduction could be a good way as a either stepping stone or as a way to help them improve their behaviors in a way that can very quickly help them in terms of their health, their well-being, and everything. So even though if we're talking about homeless people who have psychosis and have problematic cannabis use, even though the ideal thing would be treatment, it can be very difficult for these populations on top of all of the challenges that they're facing already to also go into treatment and to give that time to treatment and actually go through with it. So harm reduction in the case of somebody who's not open or willing or capable of that could actually be a very helpful alternative.
Thank you for that. There's another question here, which kind of it could be interesting in contrast to what Heba was talking about in terms of developing very cheap research to target rare diseases. Somebody commented that substance abuse treatments are notoriously expensive. So how will the harm reduction costs compare?
Well, for cannabis use disorders, treatments specifically adapted for people with psychotic disorders, there isn't yet a gold standard. There isn't yet something that's been established.
Got it.
So we can't even talk about costliness just yet. This CHAMPS, the program that we're developing, is would be once finalized, if it works, if it's proven to work through clinical trials, would be easy to disseminate and not too costly, besides the cost of like maintaining an app and updating it and everything like that, because the content is already developed and it can be self-administered. So in that sense, in terms of cost and in terms of accessibility, it's actually pretty great.
It's true, actually. If you have an app, anybody who has a phone can technically have access, which does loop back to the
Exactly.
homeless situation. If you don't have a home, it's possible you might not even have a cell phone and might not be able to access that kind of technology.
Yeah, and to add to that, so there have been surveys of people with psychotic disorders to see how much access they have to smartphones, because this was one of the main things we had to look into before even proposing such a thing,
Right.
because there is a decent proportion of people with psychotic disorders who are homeless, but it's the majority of this population that has access to a smartphone.
Oh,
So
nice.
this helps with the feasibility of this kind of intervention or program.
Got it. Is CHAMPS, this is another question from the chat, is CHAMPS for Canadian participants only?
Right now, we're still testing it. It's still in its initial testing phases. So yes, it's specifically for people in Canada that are attending early psychosis clinics in 4 different provinces. So once we figure out whether or not it works, and if it does, then we perfect it, then we test it in a full randomized controlled trial, then we'll very likely be able to open that up to more people.
Got it. So final question from the chat, somewhat related to what you're just saying. So we're in very preliminary phases right now from what you're saying. Do we have plans for data collection in terms of personalized treatment, or are we really just working on a very general level right now?
What do you mean? Sorry.
So the question specifically, are there elements or plans for data collection and personalized treatment using CHAMPS, presumably? Or is, instead of personalized treatment, is it really more of a generalized approach? Or I guess the question is, how can we personalize this?
Right, I understand. Actually, the way we developed CHAMPS was so that the participants themselves can set their own goals, and we have a lot of prompts, and it took a lot of brainstorming actually to be able to make it personalized for every person doing it, and making it using a general formula. So that was really complicated, but currently that's how it is. Currently, people who go through CHAMPS will set their own goals. They'll have their own prompts. They'll fill in their answers of what motivates them to use cannabis, to want to stop using cannabis, etc. So it is personalized until this at this moment.
By design, which is great.
Yes.
So you got all the hard questions out of the way at the beginning, and now you're releasing a product that is just by nature going to be detailed to each person.
Yeah.
That's wonderful. Thank you so much, Stephanie. This has been great. We're going to move forward. If anybody else has any more questions, like I said before, please put those in the chat. Stephanie, stick around and definitely hop in the chat to answer those if they still come. So it looks, I think there might still have questions in the chat, so feel free to hop over to those. All right. Thank you so much. So moving forward, talk number 3 of 3. I was instructed not to say best for last because all of our talks today are from the best of the best. Next up, we've got Arseny Prozorov, a master's student at University of Montreal. And his research is focused on understanding how the mechanisms of Alzheimer's disease either mirror or differ from normal, healthy forgetting. Themes in his talk include, but are not limited to, memory, forgetting, Alzheimer's, and neuroplasticity. More on that coming right at you in 3, 2, 1.
Raise your hand if you would like to have a perfect memory. Me personally, I wish I had a perfect memory, especially when I have my exams. But some of the exams, like my biochemistry exam, I was so traumatized that I can't even forget it anymore. One of the researchers from 19th century, Luria, was describing a person in his book Mnemonis who could remember every single detail of his life. He could remember everything from early childhood, what he read, what he did, and what he was saying. You think that would be perfect to have a memory like this, but actually he was miserable. He hated having this perfect memory because all the memories would be so confusing for him. They would come to his mind all the time and he wouldn't be able to suppress them. And especially the memories that he didn't want to have. And to get rid of those memories, sometimes what he did, he wrote those memories on the pieces of paper and burned them, trying to forget, but he was never able to. On the other hand, did it ever happen to you that you were looking for that word and you just couldn't remember it? Or when you were talking on your phone and you're taking your phone and you start looking for your phone, but you realize that your phone is in your hand. Or when you left your keys in that particular spot at home, but then you come there and they're not there anymore. Hopefully those are not early symptoms of Alzheimer, but actual normal processes of forgetting. And today I would like to discuss how we form, store, and forget our memories, and how forget is important for our memories as well. Hello, my name is Arseny, and I would like to present my master's project on Alzheimer's disease and neuroplasticity. So what is neuroplasticity? I'm sure most of you heard the term neuroplasticity before, and what usually it refers to is changes, physical changes in your brain to account for your experiences. And it's not just that. Brain plasticity and neuroplasticity is actually everything that you can think of yourself as a person. All your experiences, everything is encoded due and thanks to the neuroplasticity. Neuroplasticity is what makes you learn new languages, what makes you learn this piece on the piano, and how long does it take you to practice. But at the same time, there is other people who would always go to the extreme to say how extraordinary those powers could be. And we always have to take it with a grain of salt. But in general, it's been shown that neuroplasticity can be very, very helpful in our life to not just remember, but have a better mental health and brain health. So neuroplasticity. To understand how it works and how we form our memories, we have to understand a little bit how our brain regions communicate with each other. And when we experience something, when I look at the cameras and I see them right now, the experience of all of this light and everything comes in my brain and lights up certain areas in my brain. Those brain regions communicate and they form a particular trace in my brain, the unique trace that represents this particular memory. And then later on, if I come back home and I try to remember that, I actually try to access this trace again and I try to remember what happened. If we zoom in a little bit more, we can see that the connections between those regions, those are the sites where all the memory forming happens. And when we look at these connections that are called synapses, when we think about this experience, when we repeat it or we do it several times, those regions fire again and again. And when they fire several times, they get strengthened. But like when we don't maintain those memories, when we don't maintain those connections, we forget them. We can think about this as a path. If you walk on this path every day and you maintain it, you will remember it and it will become a highway easy to access. But if you don't maintain it and you don't think about it and you don't study for your exams, most likely you're going to forget it and you're not going to be able to retrieve it after. To illustrate this example, and I encourage you to try it at home as well, I'll illustrate it with my memory from my childhood. When I was a kid, when I was around 5, I remember I was in this playground. I remember the details pretty vividly. It's a vivid memory. I remember the swing behind me. I remember the fence that was around me. And I remember this metal slide with a rusty old metal slide. For me, even for me as a kid, this slide seemed like a health safety violation. But what happened in my brain at that point, when I was looking at the slide, a particular region in my brain, the visual area, lit up. And it communicated with the rest of the brain, creating this unique pattern of activity that represented this mental health violation slides. So for other parts, for other details of this memory, I still remember, for example, the touch of the sand. When I was making this sandcastle, I remember how I was touching the sand, and I remember the touch of this viscous sand that I had in my hands. But then when I try to remember it a little bit more, and when I think about this detail, I don't really know if it's the sand from a year ago when I was on the beach, or it's an actual sand touch from 20 years ago when I was a kid. And here it gets a little bit tricky because we can't really say sometimes when we try to look at those details, if those details are actually details of that memory or they overlap with others. And it makes sense biologically because for our brain it's easier to store some details and and distribute them over several memories, because it would save some energy for our brain. So this process is a part of process of forgetting, because most likely I forgot the part of the touch from 20 years ago and replaced it with the other. But to make a memory really memorable and vivid, for me, for this memory in particular, one emotional part was very important. And what happened, this kid came up to me and he wanted to play with me. He asked me to play and build a sandcastle. But instead of playing, he just stomped and he ruined it. And I was upset. And that was my emotional regions of the brain that lit up. And those emotional response, that what helped to make this memory so vivid and so easy to access right now. Okay, so from this example, we can see that memory is not a recording on a cassette. It's not set in stone. It's actually its own entity that changes and develops over time, and how forgetting some of the details is important for our brain health. But is forgetting my postal code and forgetting when you have Alzheimer's is the same, or do they have different mechanisms? And this is part of my project. That is what I'm trying to study as part of my project as a master's student. So to understand the Alzheimer's disease, first of all, we have to know the two main molecules that are considered to be crucial for progression of Alzheimer's disease. And those molecules are A-beta and tau. This A-beta accumulates in Alzheimer's brain, in Alzheimer's patients' brains 20 to 30 years before the onset of the disease. And it is considered to be one of the causative reasons. But what does it actually do? What does this molecule actually do? And right now it's believed that this molecule helps to weaken those synapses and eventually forget some of the information, deleting some of the not wanted information from our brain. So if this A-beta starts accumulating more, if there is an excess of this A-beta, it causes more and more weakening of the synapses, and eventually it leads to synapse loss, memory loss, and Alzheimer's disease. The exact reasons why there is more A-beta in some people and why A-beta starts accumulate later in life are not clear, but it seems that it's an intricate balance between strengthening and weakening of the synapses. And if this balance is broken due to several environmental or genetic factors, if this balance is broken, we have the increase of A-beta and eventually forgetting and Alzheimer's disease. Okay, so if you thought to live until 80 years and you still have 4 friends, most likely one of you would have an Alzheimer's disease. To prevent it, there is things that you can do. And actually, the recipes were known for millennia. And only right now, research is catching up with those realizations. And the recipe seems pretty simple, but it's actually very potent. It's to stay socially, intellectually, and physically active. For example, physical activity was shown to increase your level of BDNF, which is a molecule that promotes your brain plasticity and promotes growth of the new neurons in your brain. Even though I know this information, I'm not able to keep up my gym schedule. Or for example, intellectual activity. It's been shown that people with more cognitive reserve are more resistant to onset of Alzheimer's disease. So if you thought about this is exactly the time to pick up that book that you've been thinking about. Or the last but not least is stay socially active. It's been shown that people who have larger social circle are actually more resistant to have Alzheimer's disease and symptoms of forgetting, for example. So next time somebody invites you to a party, maybe it's not a good idea to say, oh, I don't feel well, and decline the offer. Maybe it's a good idea to actually go and get a bigger social circle. So party responsibly and work hard, and try to use those understandings of plasticity and Alzheimer's disease to get your brain and mental health better. Thank you so much.
Alrighty, and we're back for the third time. Thank you so much, Arseny, for that delectable talk. Very interesting. I'm excited to hop into a couple of questions right off the bat. First one we'll take from the chat. How do you differentiate between a memory lapse that occurs in a normal brain compared to one that is already under the influence of Alzheimer's disease?
This is a great question, and there is some recent research that's been trying to differentiate between forgetting in Alzheimer's and forgetting like normal forgetting. I'm not gonna go in too much into detail, but there is a term called silent synapses, which are basically synapses that are there. Is the pathways that I was talking about, they're there, but they're silent, so people are not able to access them that easily. What happens in Alzheimer, because of the at first it's just the synapses get weakened, but after the time, and especially when the tau starts accumulating, there is a death of those neurons. So what happens, that memories are actually destroyed. The memories are not there anymore at all. They don't exist. In normal forgetting, for example, memories, and there is hypothesis that a lot of, and mostly all of our memories are actually encoded in those silent synapses. Memories are still there. So this is the main difference between Alzheimer's disease, especially in the later stages, that there is actually destruction of the memories, and forgetting, normal forgetting, where it's just we're not able to access those memories that easily anymore.
When you talk about memory destruction, is that more like sending a file to the recycle bin or deleting it forever?
Memory destruction, Alzheimer's disease. So again, if we think about the engram, if we think about this network that represents memory, if we have several neurons, for example, that are destroyed, some of the other neurons can compensate, because as I said, some details can be overlapping with others, so we can try to replace those details. But if there is enough neurons that die that actually are a crucial part of that memory, then it's not there anymore. We wouldn't be able to replace it with details that are overlapping. So this is the idea. And like, to say at what point the memory that we just don't have that memory, it's pretty hard, because we're still looking at how those engram cells forms and how those networks represent the memory.
Right. The boundary of a memory is very blurry. Like you were saying, it's kind of a distributed network of fragments of a memory. So there really is no single pocket, which is very interesting. Is there, this is another question from the chat, is there any research into how disorders like ADHD interact with Alzheimer's or memory in general?
So I didn't mention that, but for forgetting, Alzheimer's, and memory, there is some disorders, for example, like PTSD, where you would have strengthening, the uncontrollable strengthening of the synapses. So people wouldn't be able to actually suppress those memories. Or OCD, for example, in people with OCD, that would be again strengthening of this compulsive and obsessive behaviors that would repeat and repeat, and that would make a highway that the person would be trying to access all the time. So yes, for ADHD, however, how ADHD would interact with Alzheimer's, that's an interesting question. I would have to think about that though.
I'm glad you brought up PTSD though, because I was personally curious. You mentioned about how A beta molecules are kind of this built-in forgetting mechanism in the brain. Could we potentially use targeted A beta treatments to eliminate traumatic memories associated with PTSD?
That'd be interesting, but the problem would be it would have to go directly into specific brain areas. For example, if we're talking about Alzheimer, or if we're talking about PTSD, that would be amygdala, which is a fear response. So we have to deliver those molecules specifically to amygdala. And it might be not a good idea, because we already have some of the techniques, which is stimulation techniques. If we, for example, give low frequency stimulation to that area, and it's been done already, we can delete that memory. So at that point, a beta treatment becomes a bit not as relevant, because we can just use stimulation treatment, for example.
Excellent. So we've already solved this problem.
I wish. I mean, that'd be great. Though.
we've at least started to attack the question of the problem. You mentioned that. So here, I'll pop you this question actually. To what degree is Alzheimer's heritable? Like if nobody in my family has it, should I be worried?
So Yeah, I mean, those are great questions, and I wish I had more time to go in a little bit more about talk about those questions a little bit more in my talk. So familial Alzheimer, 1 to 2% of population, 100% heritable, 1 to 2%. But this is heritable. This is familial Alzheimer. That's people who can be sure that most likely they'll have Alzheimer because of the certain mutations. In general, Alzheimer, we would say there is a strong environmental factor. So I mean, it's more than around 50, even more than 50%. But there is a strong genetic factor. But the genetic factor is several genes. So this is a gene-environment interaction. So several genes that have certain mutations would predispose the person to have Alzheimer's. And that is why knowing that, for example, somebody in your family have Alzheimer would help you to change a bit of, change your lifestyle, for example. And changing the lifestyle might help you not to get that Alzheimer's. So it's not a destiny, but it could help you, knowing that it could help you to prevent it.
I also love that you provided lots of examples towards the end of your talk of how we can try and combat the development of Alzheimer's, or at least kind of mitigate some of its effects as we get into older age, and not even in older age. I look forward to being 80 and to having lots of friends, hopefully. Hopefully more than 4, so that I can lessen the chances of me being the one. So that's awesome. Arseny, thank you so much. Stick around. There's plenty more questions for you in the chat. And I would love to also engage more with all the speakers today, because there's still many, many unanswered questions. But that's the beauty about sharing research, is we can develop lots of questions without being able to answer all of them right now. So thank you very much, Arseny.
Thank you.
For those of you who have not put any questions in the chat yet, please do so. It's not too late. Questions for any of the speakers. Still opportunities to win those great prizes that we've got for you. Amazing. What a treat. Great way to start the morning off and start the long weekend for those of you who live on Earth. Thank you once again for joining NeuroLingo today. Absolute pleasure to have you. And thank you and congratulations to Hiba, Stephanie, and Arseny. Woo! Special thanks to the speaker trainers as well, Dr. Edward Ruthazer, Dr. David Ragsdale, Dr. Marion Van Horn, Nicholas Belvaux, Michel Rochon, and Dr. Natalie Arbour for coaching our speakers for the past 3 months. Thank you so much to them. Today's event was supported by Tannenbaum Open Science Institute at the Montreal Neurological Institute. It was also supported by the Integrated Program in Neuroscience, IPN, at McGill University, the Postgraduate Students Society of McGill University, and SCHUM Research Center, as well as the Douglas Mental Health Institute. Finally, we would like to extend our gratitude to those of you who donated during the event registration. Thank you so much for that. For those of you who are curious as well, the presentations will be published on YouTube in the coming weeks. So in case you popped in late or you sneezed and you missed something, there's definitely an opportunity to rewatch and revisit the memories that you formed and really cut that past so you remember this forever. At 1 p.m. today, there will be a French-speaking event featuring Hiba and other master's students from McGill University discussing the topic of neurogenesis. So definitely pop back in at 1 p.m. for that. Link to register will be in the description. As well, we ask that you please fill out the survey linked in the chat and the description of the YouTube video to let us know how we did today and how we can improve in the next event, because there will be many more of these for you. So goodbye for now. Stay safe. Take care. I'm Jeremy Allman, and have a lovely rest of the day.